Hello everyone, how are you all doing? This is your Tithi ma'am and I'm going to discuss the CSIR June 2022 question paper, morning shift paper for Unit 8. Are you all excited? Ready? Okay, be ready with your pen and paper so that you can note down the explanations and whatever we are going to discuss here. So this time in the morning shift paper, we saw around 10 questions total from Unit 8, that is Inheritance Biology. Out of those 10 questions, there were around 5 questions from Part B and 5 questions from Part C. So that is what we are going to discuss here in this video. So starting with it, first question, which was from Part B, which one of the following condition represents autopolyploidy? Okay, autopolyploidy. Now, whenever we talk about autopolyploid, so it basically we are talking about the euploidy situation. Polyploids means euploids. Euploids are the mutations where one entire chromosomal, entire haploid set of chromosomes is added or deleted. Okay. Now, the word auto always refers to self. Okay. So, the added set of chromosomes that we are doing here in the genome, when it belongs to the same parental species, the self-parental species, that is when we call it as autopolyploid. Okay. So, let's see the option. Which of the following condition is correct for autopolyploid? First says more than 2 set of chromosomes, both of which are from the same parental species. Yes, same parental species. Okay, let's see the other options as well. So option 2 says more than 2 set of chromosomes, both of which are from the different parental species. Third, more than 2 set of chromosomes only from a single parent. Fourth, duplication of a chromosomal locus leading to spontaneous increase in the copy number of a gene. No. So, autopolyploid will be more than 2 sets of chromosomes, both of which are from the same parental species. Okay, so that is what is called as autopolyploidy. The other one from different parental species, it's called as allopolyploidy. Okay, so answer will be option 1. So you can see the explanation here that autopolyploidy appears when an individual has more than 2 sets of chromosomes, both of which from the same parental species. Okay. Autopolyploidy results from accidents of meiosis or mitosis and produce extra set of chromosomes, all derived from a single species. Non-disjunction of all chromosomes in mitosis in an early to an embryo, for example, doubles the chromosome number and produces an autotetraploid because it belongs to the same species. Okay. Alternately, triploids may arise from cross between a autotetraploid that produce 2n gamete with a normal n gamete. So that will result into 3n. Okay. So anyways, autopolyploid, auto means self. So autopolyploidy will mean that the extra set of chromosomes are added from the same individual, same parental species. Okay. So this is what the autopolyploidy looks like. Okay. So if you have a normal diploid chromosome here, okay, normal diploid cell here. So it has each chromosome of 2 types. So autopolyploid means that the same parental species chromosomes are added. One set is added. So now all the chromosomes are present in a group of 3 or here a group of 4 like that. So it will be autotriploid or this one will be autotetraploid. Okay. Let's move on to the next question. The following picture represents a gel profile of a pair of DNA markers observed in parents P1 and P2 their F1 progeny and F2 progeny. 4 different profiles were observed in case of F2. The number of F2 progeny showing a given profile is indicated in the brackets. Okay, so the number is given here. So in that, basically it means that out of F2, 98 individuals are showing this kind of profile, 28 individuals are showing this profile, 33 this profile, and 11 this profile. Okay, now based on the above observation, which one of the following statement is correct? So here it is not a question which is solely based on unit 8, but you can say that it is also related to unit 13 as well, where we talk about the markers, the DNA markers. Okay, so first statement says that codominant DNA markers were used in this study. Now what are codominant markers? Codominant markers means they will show their presence, okay, together. Even when they are present together, that is in heterozygous condition, they will show a profile that will be different from either of the homozygotes. Okay, so codominant DNA markers, they will always show their expression. They will always show some or the other profile. Okay, it won't be completely absent. The bands will not be completely absent. The main difference will be here that the heterozygotes profile will be for sure different from the homozygotes. Okay, now if we see here for codominant DNA marker, okay, till all this it is fine, it might be codominant, but what about this progeny? In this progeny in F2, none of the bands are showing. Okay, that is a thing which is not possible for codominant DNA markers, so this statement is not possible. Okay, now the polymorphic DNA bands represent 2 independent genes. Now how to find it out? Now you see the ratio here in F2. In F2, what you have? We have a ratio of 93 is to 28 is to 33 is to 11. Okay, now this ratio, if you see it, is almost same as 9 is to 3 is to 3 is to 1. And what kind of ratio is this? You can recall this ratio, right? This is a dihybrid ratio. A ratio of a dihybrid cross, correct? The phenotypic ratio of a dihybrid cross is what it looks like, correct? So that means, yes, there are 2 different genes involved. Dihybrid cross means a cross where we study the inheritance pattern of 2 different genes. So yes, there are 2 different genes involved here. So polymorphic DNA bands, it represents 2 independent genes. Yes, this might be correct. Okay, now let's check out the other options as well. Okay, so if there are 2 independent genes, okay, so let's say that the 2 genes that we are talking about, A and B. So if this band is for gene A and this band is for gene B, okay, so the capital alleles are showing the band. Okay, so if this individual is having this band here, means it's the P1's genotype will be what? Capital A, and small b, small b. Okay, it does not have the capital B allele, that's why the other band is absent. In P2, just the reverse, small a, capital B, capital B. Okay, where this gene has a dominant allele capital A and a recessive allele small a. Similarly, this gene B has a dominant allele capital B and a recessive allele small b. Okay, so P1, P2, they are having this genotype. So what about F1? If you cross them, F1 will be a heterozygote. Okay, so it has 1 capital A allele, 1 capital B allele. That is the reason F1 is showing 2 different bands. Okay, now what it is saying, if P1 parent, so what is P1? This is crossed with F1 individual, the progeny will show all 4 profiles as observed in the cross of F2 progeny. Okay, so in F2 progeny, basically what all genotypes are we seeing? So in F2, this is 9:16 ratio, the first column. So 9:16 ratio means both dominant are present. So capital A dash, capital B dash, right? That is our 93. Next, which one it is there? It is only capital A present, right? So capital A dash small b. This is 28. The third variety, only the B band. So small a, capital B dash. So this is 33. And the last variety is none of the bands are present. That means both the genes are present in recessive state. So homozygous recessive, right? Now, when we do the cross between this individual and this individual, what we will get? So P1 cross with F1. So P1 was capital A, small b crossed with a double heterozygote. Okay. So you can make the Punnett square very easy. This individual will form only one type of gamete, capital A, small b. This individual will form 4 different gametes, capital A, capital A, small a, capital B, and small a, small b. Okay. So what all progenies are possible here? One progeny, second, third, and the fourth variety. Okay. Now if you see here, okay, so dominant, one dominant allele, one dominant allele, it is present. Okay. So the first one, that is capital A dash, capital B dash, that variety is present. Okay. So here also, this one also. What about this? Here capital A dash and small b, this variety present out of F2. Last one, capital A dash, small b, again the same. Okay. But what about the other two? That is small a, capital B dash, or small a, small b. These two varieties are missing here. Okay. So when we do the cross between P1 and F1, all the individuals showing the profile in F2 cannot be formed. Okay. That's why this statement again is wrong. The fourth one, if an F2 progeny which does not show either of the DNA marker, that means we are talking about the homozygous recessive, is crossed to a P1 parent, means this one, the obtained progeny will have 2 types of individual. One which shows a band and other one with no band. Okay, so let's just do the cross. So again it will be the last variety, that is small a, small b is crossed with P1. Okay. Now again here, what do we see? This individual will produce only one type of gamete. This individual will produce only one type of gamete. So their cross will have only one type of individual. Only one type of progeny, not 2 types. Right? So this one will have only one band. There is no other progeny formed. So the other progeny with no band is shown. That we are not even getting here. So again, this statement, fourth statement, is wrong. Okay. So we were correct with our interpretation. That answer is the correct answer. According to the profile that is given to us, the answer is option 2. That is the correct statement. Okay. So statement 2 is correct, that as the number of F2 progeny is like the ratio 9:3:3:1, which means genes are assorted independently. Thus polymorphic DNA represents 2 independent genes, A and B. So F2 progeny is this varieties. Okay. All other statement 1, 3 and 4 are incorrect. Moving on to the third question from part B. The additive nature of genetic map as suggested by Alfred Sturtevant and T.H. Morgan is possible if there is no interference in crossover, complete interference in crossover, partial interference in crossover, variable interference in crossover dependent on the genetic distance. Now for that, let's first understand what is additive nature of genetic map. Okay, so suppose this is a DNA molecule and we are talking about 3 genes placed on it. Okay, A, and B and C. Okay. So additive nature, what it says is that if we find out that the distance between AB is 4 centimorgan and distance between BC is 2 centimorgan, then we should also find out that the distance between A and C will be 4 plus 2, that is equal to 6 centimorgan. Okay, that is what is the additive nature. Now, that additive nature is followed if there is no crossing over, and that was found by T.H. Morgan and Sturtevant by their experiment on Drosophila, that this addition is not followed when we are talking about large chromosomal distance. Okay, now why this is not followed? The major reason behind it is the double crossover that happens. Okay, now see what happens because of the double crossover is, so suppose you have this A, B and C, and you have another homologous DNA with small a, small b, small c. Okay, so when the distance between this three set of genes is quite large, then the physical distance between each pair of gene increases, and because of that there is a chance of double crossover, means one crossing over event happening here between AB, one crossing over event happening between B and C. Right? Now, if that is the scenario, then the double crossovers will be, what will be the situation? They will come as A, small b, capital C, or small a, capital B, capital C. Right? Now, when we are considering three different genes, we can differentiate the double crossovers from the parentals. Parentals are capital A, capital B, capital C, small a, small b, small c. Okay, but when you are considering just the terminal genes, just consider A and C. Okay, so in the after double crossover also, the A and C's will appear as capital A, capital C and small a, small c. Okay, so we will not be able to differentiate the double crossovers from the parental types, and therefore we will count more parental types, and more parental types will mean closer the distance are, closer the genes are placed together on the chromosome, means stronger is their linkage. But actual scenario is just the opposite. So genes are very far apart, and that's why double crossing overs are happening. And because of that, the simple addition that if this much is 4 and this much is 2, then A and C will be 6 will not be followed. Okay, so distance in 2-point test cross between A and C, the distance will come smaller. They will come more linked, but which is not actually true. Alright, so why there is this discrimination? Why this additive nature of the genetic map is not followed over larger distance? Because of double crossovers. Okay, so if we want to follow this additive nature, what we have to do? We have to make this double crossover 0. That means no double crossover should happen. Right? Now, what is the phenomena that decides whether double crossover is happening or not? That phenomena is called as interference. Okay, so if I is equal to 1, if total interference is there, that means double crossover will be 0. Right? 0 double crossover will happen, and if that happens, then the additive nature of the genetic map will be followed. Right? So this assumption is not followed over long chromosomal distance according to Sturtevant's data, and the reason behind it is double crossover. Now, since interference is what? It interferes with the probability of double crossover. So basically, one chiasmata formation between the same pair of chromatids will stop the chance of another chiasmata forming. Okay, so if interference is complete, then no two double crossover will happen. Okay, and if there is no double crossover, then the additive nature of the genetic map can be possible. So answer here will be option 2, that complete interference. So if we want to, the additive nature of the genetic map can be possible if there is complete interference in crossing over. Okay, so this question is not the usual kind of question that is asked, but still, as we always say that this genetic map is very, very important. So you get a question from genetic map, and this question is more based on the concept. Okay, how well you have understood the concept of interference. Next question coming from microbial genetics and microbial genetics related questions. It has always been popular, but from last 2-3 times this has become even more popular. Okay, so in a conjugation experiment between bacterial HFR strain X and F- cell, lac gene enters the recipient in 4 minutes, but the F- cells remain auxotropic for leucine, TRP, URA, GLU, PHE and gly. The mating is then allowed to proceed for 20 minutes and lac plus X conjugates are selected. So of the lac plus cells, okay, the percentage of cell who are showing that particular gene marker is mentioned. So select the correct order of the genes as they enter from the choices given below. So the gene order is given and you have to see which one is the correct order. See, now let's go to the basic here. What is conjugation? Conjugation is exchange of genetic material between two bacterial cells via a conjugation tube. Okay, a physical contact is required. Okay, now in conjugation what will happen? So as long as physical contact is there, the DNA will be transferred. Okay, if the physical contact breaks, the transfer will stop. Okay, now so the transfer started with lac gene, right? So lac should be the first one moving out. So after the F plasmid, after the origin of transfer of the F plasmid, it should be the lac gene. Okay, so yes, lac. So we can eliminate A and C because lac is not the first one. Okay, so out of B and D. Now everybody will be lac, so it is not given because we are only selecting the lac plus genes. Okay, now so the longer, so for conjugation, what is required? That the conjugation tube remain intact. But this conjugation tube, the chance of it to dissolve is more as the time increases. Okay, because more the time, then the chance of conjugation tube is dissolving is more, because it becomes difficult for the bacteria to stay in contact. Okay, so the farther the genes are away from the origin of transfer, lesser will be the chance of getting a recombinant of that particular marker, because lesser is the chance that the marker will be transferred from the HFR cell to the F- cell, right? And closer the genes are to the origin of transfer, more is its chance to get transferred. Okay, so what do we see here? TRP is 98%, right? The next highest frequency. That means this TRP plus has to be right after lac. So after lac, it is TRP. Okay, then after TRP, so we have got lac plus first. After lac, it has to be TRP. Okay, now after TRP, the next highest percentage is what? GLY. Okay, so it has to be GLY. After GLY, next highest percentage, GLU. So it is GLU. After that, next highest percentage, LEU. And then URA. And finally, 0% is PHE. That means it has to be the gene which is located farthest from the origin of transfer. So in this order, if we have to write down the genes, then this should be the order: lac, TRP, gly, GLU, LEU, URA, PHE. Okay, so answer will be option D. Very easy, basic interrupted mating experiment from conjugation. Yes, now this time we got a pedigree question. So given below is the pedigree indicating the pattern of inheritance. Which one of the following options correctly describe the pattern of inheritance shown in the above pedigree? Okay, so options are this four. Right. Now, I always tell you in the class that when you are doing the pedigree analysis, always take it like a crime investigation. Okay, so you start with all the possible suspects, start eliminating the suspects depending on what kind of evidence you are getting from the pedigree chart, and then you choose the inheritance pattern with which you are left at the end. Okay, so we will start with all the possible inheritance patterns. Now, what is the characteristic for a dominant inheritance pattern? Okay, so for any dominant inheritance pattern, any trait that is dominant because of the dominant allele, if the individual has it, that means the individual has got the allele from either one of the parent. Okay, so if the parent has it, because it's a dominant allele, so the parent will also be showing the trait, right? Now that is what is not happening here, because here both the individuals of the second generation are affected, but none of their parents are affected. Okay, that means it cannot be a dominant trait. Okay, it cannot be X-linked dominant or autosomal dominant. So you can simply eliminate them. Now next one, we are talking about X-linked recessive or autosomal recessive. Okay, so let's check for X-linked recessive. Now if the female here, if the male and female, they both are affected, right? So if they both are affected, so suppose we are talking about X-linked, right? So in X-linked, if the female is affected, means she has, so if let's say that this is the gene which is present on the X chromosome, so the recessive allele is small x, dominant allele is capital X. Okay, we are checking whether it can be X-linked recessive or not. So in that situation, so if the female is affected, means she has two copies of small x, right? She is homozygous recessive. One small x allele, fine, she is getting from her mother. Okay, now other small x allele she has to get from her father. That means that makes her father small x capital Y, and if her father is small x capital Y, her father also has to be affected if the disorder is because of the recessive allele. But since her father is not affected, right, that means it cannot be the X-linked recessive. It has to be autosomal recessive, and that is when it is possible that this individual can be small a, this individual can also be small a. Let's suppose that if it is gene A and the recessive allele is the causative allele. Okay, then these two individuals can be heterozygotes, and that's when it is possible. Okay, so X-linked recessive is also not possible. Only possibility is autosomal recessive. Now this question, if you see, it is quite easy compared to the other kind of question or probability related question that are usually asked in the CSIR paper. So a trait cannot be X-linked recessive as well because for the daughter to be affected with an X-linked recessive trait, she must receive the X-linked recessive allele from both father and mother. And as the father is not affected, it means he does not have the X-linked recessive allele, and that's why it cannot be X-linked. So the last option that we are left with is that it has to be the autosomal recessive. Now that's all from the part C from unit 8. Now we are moving on to, that was all from part B from unit 8, and now we are moving on to part C questions. So the first question in part C, In Drosophila, balancer chromosome are used to keep all the alleles on one chromosome together. A balancer contains multiple inversions so that when it recombines with the corresponding wild type chromosome, no viable crossover products are formed. Balancers also carry an allele for a dominant phenotype. A Drosophila male with sepia eye color is crossed to a female containing a third chromosome balancer. Okay, so we will write down. So for eye color is the trait we are talking about. Okay, so one it's calling the sepia. Now the allele for sepia phenotype is located on chromosome number 3 and is recessive to the wild type eye color. So sepia is recessive. The dominant marker on TM6B, TM6B is the balancer chromosome which is mentioned here, is a tubby phenotype. Okay, so eye color tubby, that is dominant. Mentioned that is the dominant marker. So further, an individual homozygous for TM6B balancer does not survive. Now, F1 progeny with tubby phenotype is submitted. The F2 progeny is expected to have which one of the sentence will be correct. Okay. Now, for that, let's talk about this eye color. So, just for our easier crossing, let's say that the allele SE is responsible for sepia, small SE because it is recessive, and capital S is for tubby eye color. Okay, so basically eye color has now two alleles. Small SE and capital S. So capital S is dominant and forms the tubby eye color, and SE is recessive and forms the sepia. Okay, now, so what is the cross mentioned here? So, it is mentioned that a Drosophila male with sepia eye color is crossed to a female carrying the balancer chromosome. Balancer chromosome has the tubby. Remember? Yes. So, what is the cross? Cross is male is sepia. Okay, so it is homozygous for sepia crossed with female. Okay, who has one balancer chromosome. Capital S, we are leaving the other allele as blank because we know that she is stubby. So she will have one capital S. She has one balancer chromosome. Other allele we don't know. We are leaving it blank. Now in F1 generation. So which F1 we have taken? We have taken the F1 showing the tubby phenotype. Okay. So here capital SK is coming from the female. Small SE will come from the male. Okay. So this tubby phenotype individual in F1 has to be heterozygous. Now when it is becoming heterozygous here, now next sentence it says that F1 is submated. Submated means F1 is self-fertilized. Okay. So SSE is self-fertilized with SSE. Right. Now very easy, you can make the Punnett square here. So this produces two alleles, this produces two alleles. Right? Now what is their proportion? So capital S will be 1 by 4. S, capital S, SE will be 2 by 4 and SE, SE will be 1 by 4. Right? Now it is clearly mentioned in the question that whoever is homozygous for the balancer phenotype, they will not survive. Okay. So this individual has tubby. Why? Because it has got both the copies of this one and this one who are both present on the balancer chromosome has come together. So this individual will die. Okay. So who are we left with in the F2 progeny? So F2 progeny will have tubby and sepia in the ratio of 2:1. So in F2 generation, tubby is to sepia will be there in the ratio of 2 is. Okay. So only sepia eye color will be seen in the F2 progeny. Wrong. Sepia, tubby and wild type flies in the ratio of 1:2:1. Wrong, because homozygous for tubby will die. Sepia and tubby flies will be seen in the ratio of 1:2. Yes. Sepia and wild type flies in 3:1. That is again wrong. So this is the correct answer. Okay. So this is what is mentioned here. So the F1 cross, the F2 progeny, this proportion will die, will not survive. So sepia is to tubby 1:2. Moving on to the second question. So an individual is heterozygous for the reciprocal translocation as shown below in the given diagram. So the reciprocal translocation is given where a segment of chromosome has been transferred from N1 to N2. Okay, N1 and 1 and 2 are the two chromosome. N1 and N2 are the normal copies of the chromosome. T1 and T2 are the copies which has undergone the crossing over. Okay, so EFG, not crossing over, sorry, the translocation. We are talking about translocation. So it has undergone the reciprocal translocation. So EFG has been transferred from 1 to 2 and QRST has been transferred from 2 to 1. Okay. Now the following statements are made about the segregation of each chromosome during meiosis and gamete formation. First, the complexly paired 4 chromosome fail to segregate, pass into one cell at anaphase 1 and the cell eventually dies. Now to answer this question here, let's see what happens in the reciprocal translocation. So this is the image of reciprocal translocation. So in times of reciprocal translocation, at the time of meiosis, the translocated chromosomes, okay, they will form because they will have some homology with N1 and some homology with N2. So they will align with both of them. And because of that, this plus kind of structure will be formed. Okay, so alignment will be there. So statement 1 says that they will fail to segregate. That will not happen. In fact, these 4 chromosomes, they can segregate into the gametes in 3 different ways. Okay, so statement A wrong. B, the chromosome pair between the regions of maximum homology keeping the translocated part unpaired and normal meiosis occur. That is also wrong because the translocated part as well as the non-translocated part, they all will pair. Okay, so the non-translocated part will pair with its regular homologous member. The translocated pair will pair with the other chromosome, the non-homologous chromosome with which it has exchanged its parts. See, one of the way of chromosome segregate is by alternate segregation where N1, N2 move to one pole, N1 and T1, T2 move to another pole. Now that is absolutely correct. You can see alternate segregation, which means N1 and N2 will move to one pole and T1 and T2, they will move to another pole. Okay. Now the gametes which are receiving this alternate segregation, then they will be completely viable. Why they will be viable? Because all the genes are present in them in equal amount. Okay, so A, B, C, D, E, F is also present and JKL, GH, I, J, K, L is also present. Okay, now this one also, the genes, all the genes are present. The position of the genes are different now. Okay, but the genes are present and that's why the gametes will be viable in alternate segregation. So this statement C is absolutely correct. D says that alternate segregation produces non-viable gamete. Now that is wrong because alternate segregation will always produce viable gametes. The other two mode of segregation, that is adjacent 1 and adjacent 2 segregation, will produce non-viable gametes. Now E statement says that reciprocal translocation are considered as crossover suppressors as no gametes with crossover products are produced. Okay, now that is not true. Inversion is considered as the crossover suppressor, because then it becomes difficult for the chromosomes to align with each other. But when we are talking about reciprocal translocation, they can align with the chromosomes very properly. Okay, the original one as well as the translocated one. Right? So E is wrong, only statement C is correct. So answer will be option. Moving to the third question from part C. The following represents the sequence of different alleles of a gene found in family represented by mother, father and their two sons, son 1 and son 2. Further, a new mutation is observed in one of the alleles of the son, which is marked with a triangle. Okay, so this is the mutation, that is C. Now, the following statements are made about the mutation. The mutation arose in the germline of father. The mutation arose in the son. The given DNA sequences are present on the X chromosome. There is possibility to use RFLP for tracking this variation. So let's talk about whether the mutation, so let's check this A and B, whether it arose in the son or whether it arose in the father. Now see, son 1 allele 2 has the same mutation just like son 2's allele 2. Okay. Now since they both have the exact same mutation at the same position, then it is highly unlikely that they both occurred separately in both the sons. Okay, the more probable is that they inherited it one from one of the parents. Okay, so mutation arose in the son, not possible. Now, did they get it from the father or from the mother? Let's check. So here, what do we see? Now, when you compare the sequence here, okay, so you will see that this sequence matches exactly with that of the father's allele 2. Okay, if you skip this position, apart from that, all the sequence is matching here. Okay, so which means that this allele 2, son 1 and 2 has inherited from the father. Okay, and that means the mutation has arisen in the germline of the father, and that is why it has got passed on to both the sons. Okay, so A is right. Now, the given DNA sequence are present on the X chromosome. How to check that? Very simple. Okay, see, the X chromosome, if it is present, then all the genes in the males present on X chromosome are present in a single copy. Right? They have to be present in a single copy because the males have one X and one Y chromosome. Okay, but since here the father is a male, son is a male, son 2 is a male, okay, and all of them have 2 copies of the gene. That means it is not present on an X chromosome, it has to be present on an autosome. Right? So that's why statement C is wrong. Now D, there is a possibility to use RFLP for tracking the variation. Now that is correct because there is a change in a single site, right? Single nucleotide is changed here, is substituted here. Now because of that single nucleotide change, if there is a formation of a new restriction site or if the existing restriction site is lost, then the RFLP, the restriction fragment that will be formed by using that particular restriction enzyme, it will change. Okay, so the RFLP pattern, that is restriction fragment length polymorphism, will be seen. So this variation can be tracked by RFLP. So statement D is correct. So correct answer is option 2, A and D. Now next question, another popular question. It has been quite popular, although last 2 sessions, last session actually, no question came from this part, which is about the mutation reversal study. But this time it came and it is very, very easy. So 2 new chemical compounds X and Y are synthesized in a laboratory and tested for their potency as a mutagen. The nature of the mutation produced by this compound is tested for reversal by other known mutagens, and the following results were obtained. Which statement best describes the nature of the two mutagen? Okay, so first of all, let's see, fibromouracil and ethyl methane sulfonate and hydroxylamine. Okay, they all produce transition. Okay, hydroxylamine, it is not reversed. X and Y, they both are not reversed by hydroxylamine, whereas X is getting reversed by fibromouracil. So it is happening transition. But hydroxylamine, what it does, it can only do transition in a particular direction. It cannot do in both the ways. Right? That's why maybe it is not reversed by hydroxylamine. Acrid in orange, it does only one type of mutation, that is insertion or deletion of single nucleotide. Okay, so X causes transition, Y will cause either insertion or deletion. Now see the options here. So option 1 says that compound X produces single base substitution to generate CG to TA transition. Compound Y produces insertion or deletion. This is our one contender. B says that X produces insertion deletion, Y produces transition. Wrong. C states that option 3 states that X produces transition, Y produces transition. Wrong. Y produces insertion or deletion. Option 4 statement, compound X produces substitution that generate insertion. Y produces deletion. Now X is not producing insertion, it is producing transition. Okay, so this is also wrong. Answer will be option 1. Moving on to the next question from part C. Again, a question coming from pedigree inheritance or pedigree analysis. Given below is a pedigree indicating a pattern of inheritance. The following statements are drawn from the above pedigree towards the understanding the pattern of inheritance. Okay. Now this question, if I see, is very, very easy, because here you don't have to conclude what kind of inheritance is there. It is just talking about the patterns that you can see in the chart. Okay, which you can just analyze and comprehend by watching the by studying the chart. Okay, now in the pedigree, so we know that when it is empty square or circle, that means the individual is not affected. If it is complete square, complete black square or black circle, that means the individual is affected. But when there is a dot inside, means the individual is carrier. Okay, so this one, this, they all are carriers. Now, statement A says that affected male does not appear to pass the trait to his sons. Now, that is absolutely correct. The male is affected, but none of the sons affected. Male affected, none of the sons affected, right? So, the affected male is not passing it to the sons. So, statement A, correct. B, an affected male appears to pass the allele to the daughter who is unaffected. Yes, that is also right, because when you see this male affected, but he is passing it to the daughter, and daughter is becoming a carrier. Daughter is not affected, but he is passing it to the daughter. Okay, here also it's passing to both the daughters, right? So again, statement B is correct. Now D, all affected individuals have at least one affected parent. Now that is not true. This individual is affected, none of the parents are affected. Affected individual, none of the parents affected. Okay, so nowhere it is happening. So this statement wrong. D, the given trait appear to be a recessive one. Now that is going to be true. Why? Very simple, because carrier is only possible for recessive trait. Carrier is somebody who has the positive allele but still not showing the phenotype, and that is only possible when in case of recessive alleles, which can stay in the hidden form in the heterozygote, right? So yes, the trait is a recessive one. E, the given trait appears to be autosomal recessive. Now that is wrong. Why? Because it is showing a crisscross pattern of inheritance. As you can see here, that the mother is carrier, but the son is affected. Okay. Now here the mother is carrier, son is affected. All right. Now the father is affected and he is passing the allele, the causative allele, to the daughters here, right? So daughter is a carrier, it's coming, and it is only possible when it is passing the capital, the lower case x. Okay, when it is passing to the recessive allele, the recessive X-linked allele is passing to the daughter. That's when the daughter is becoming carrier. Alright, so it is not autosomal, it has to be X-linked because it is showing the crisscross pattern of inheritance. So which all statements are correct? A, B and D only. Answer is option 2. Alright, so as Nelson Mandela said, that winner is a dreamer who never gives up. So don't give up. Keep trying, keep practicing. If not this time, I completely wish that you all qualify this time. But if you are not, then don't disappoint. Don't leave your weapons. Don't stop your preparation. Keep doing it. And next CSIR is going to be your CSIR. Thank you everyone. Stay tuned to Biotecnika for all other discussions on other topics of CSIR net question paper. Thank you everybody.